This is Laura Fulford, welcoming you to this bite-sized bio web seminar, which today is sponsored by Leica. Leica Microsystems is a market leader in microscopy, providing wide-field, stereo and confocal solutions specifically for biological research. This year, Leica proudly celebrates 10 years of pioneering super-resolution innovation. Leica are committed to educational initiatives that provide application and technical know-how through media such as webinars and via the Leica Science Lab Knowledge Portal. Today's presentation is titled Basics of Confocal Microscopy and is being presented by Dr. Alison Dunn, the Facility Manager of the Edinburgh Super Resolution Imaging Consortium. This is at Heriot-Watt University in Edinburgh. She graduated from the University of Sussex in 2007 with a degree in molecular medicine, after which she worked at Unipath for a year in prenatal diagnostics before beginning her PhD at the University of Edinburgh. During her PhD, she used a range of microscopy techniques to study membrane dynamics, specifically exocytosis. In February 2013, she worked with her supervisor to establish the Edinburgh Super Resolution Imaging Consortium, which provides a platform for researchers to access super resolution microscopes. She still carries out some of her own research, but primarily trains new users, works with users to develop their projects, organize events, and manage the admin and outreach. As always, we will have a question and answer session after a presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Alison at the end. So now over to you, Alison, for the presentation.
Hi, my name's Ali Dunn. So in this webinar, I'm going to go through the basics of laser scanning confocal microscopy. I'll run through the theory, and then we're going to have a work through the software so you can see how it works in application. Confocal laser scanning microscopy is a technique often favoured by biologists, as the images produced are much sharper and they allow you to image thick biological sections without losing image quality. So you can see this in the example here. So this is a kidney section and we have it underneath wide field or epifluorescence illumination and also under confocal illumination. And as you can see, the confocal is a lot sharper. So a fluorescence microscope, as the name suggests, is based on fluorescence. And this is where a molecule absorbs light of one wavelength and emits light of a longer wavelength or of lower energy. So GFP is the most well-known fluorescent protein. So if we have GFP tagged to our protein of interest that's inside a cell, I've got an example here which is a HEC293 cell with actin, which is part of the cytoskeleton. If we illuminate this with a lamp source, then we can collect the light using a camera, and this is generally how your basic epifluorescence microscope works. The best way to explain how a confocal microscope works is to compare it to an epimicroscope. So there are some important aspects to note in the system. First one is that the entire sample is illuminated by the lamp source, and the second is that the image is acquired just like a camera. So that means the whole field of view is going to be recorded at once onto a grid of pixels. So there's a limitation with this method in that everything in the cell is illuminated and all of that emitted light is going to be collected. And that includes the light that is out of focus. What is out of focus light and why is it a problem? So if you were to take a photo with your camera phone, you can choose to focus on a certain object. So in this example, I've taken a photo of the Leica control knobs and I've chosen to focus on the second one in. As you can see from this picture, that knob is nice and clear, whereas the ones that are below and above it are blurry. And this is because they are above and below the focal plane, and so they are out of focus. So if you imagine a cell on a glass cover slip, and here the red line is representing the focal plane, which is where you want to image, any light coming from this plane is going to be in focus, and it will create a nice sharp image. Whereas the blue lines are out of focus, as they are above and below the focal plane, and light coming from these planes will therefore be out of focus, and it will create a blurry image. So in an epifluorescence microscope, all of this light is going to be collected. Why is this a problem when we're looking at biological samples? Well, if our sample was very thin, 2D structure, then it wouldn't be a big issue. But usually with our samples, we're not looking in 2D. Often our structure is 3D. We're looking at something like a cell. And our cell is going to be filled with fluorophores. It's often going to be molecules inside our cell which are going to autofluoresce. And there are also going to be liquid-filled compartments which are going to scatter the light. So all of these conditions are going to cause fluorescence outside of the focal plane and consequently then blurring and degradation of the image. So if we go back to the original example I showed you, you can see what I mean by degradation, as the information you receive is degraded and it's lost. Looking at the confocal microscope, there are a few key changes which can combat these issues. First one is that the sample is illuminated by a laser, which is going to be focused to a small spot. Secondly, there is a pinhole in place, which is in front of the detector. So how do these things actually help? The focusing of the laser at the focal plane means that at the focal plane you have excitation by a small intense spot of light, whereas the planes that are above and below receive much lower intensity of light. And what this does is it gives you a reduced signal in the outer focus planes. And then we have the pinhole. So this is a slit that goes in front of the detector, and it only allows in-focus light to be detected. So the majority of outer focus light is going to be rejected, and it's not included in your final image. Okay, so now going on to the laser scanning part. Often what we want to do is image a cell or a cellular compartment, which can range between 5 microns to 20 microns. But our laser spot is only hundreds of nanometers in size, and this is thousands of times smaller than the area that we actually want to image. So how are you going to image it? One option would be to move the cell, but the other option, and the much easier option, is to move the laser across the sample. This can be shown very simply with a compact mirror and a laser pointer. So by shining a laser onto a mirror and moving that mirror from side to side, you can direct a laser to scan an area. This is what the scan head on a confocal does. It uses motorized mirrors to scan the laser across your sample. So because the laser is scanned, the collection of light has to be different. So you couldn't really use a camera in this setup, so you have to use things called detectors. Specifically with a confocal, you use detectors which are called PMTs, or photon multiplier tubes. So what happens is the laser moves along the image, and it moves along pixel by pixel, and at each pixel the light is collected and sent to the detector. Okay, so a photon multiplier tube does exactly what it says. So it collects photons and it amplifies their signal. So it creates a digital signal that can then be processed by a computer which generates your image. And it does this pixel by pixel. So when is it good to use a confocal? The biggest advantage of confocal is optical sectioning. When you're interested in whole tissues, these can often be very thick, and you often have to physically section them using a microtome to be able to get a clear image on the microscope. So this isn't always ideal, as it can physically alter the tissue structure, but what confocal laser scanning microscopy allows you to do is optically section, so you can acquire clear images from thin sections within a thick sample without any need for physical sectioning or for post-image processing. And this is due to all those key adaptations that I've talked about to having the pinhole and using a laser. There are other advantages to using a confocal, but we can go through these in the software. So lastly, just want to outline the main disadvantage of confocal, which is the speed. If you did want to image something that was happening very quickly, so processes on, say, the millisecond timescale, then you wouldn't really use a confocal, you'd use an epifluorescence microscope. And this is because a confocal is scanning, so it's going to be a lot slower. And also the longer that you are illuminating your sample, the worse the photobleaching is going to be. With confocal, you can get photobleaching and phototoxic effects. So if you had a sample that was quite photosensitive, it would be better imaged on a wide field microscope. As we've already said, epifluorescence does include all of that outer focus light. So if you are going to use a wide field or epifluorescence scope, then it is quite good to use deconvolution. It's not a necessity, but it does help. And what this does is it restores the image. And it does this by reallocating the outer focus light back to where it thinks it should be. To do this, you do need to use specific software, and you do need to acquire your images correctly so that the analysis is then done correctly. We're going to move on to the software now. So the good thing about the Leica software is it's very visual, so it's really good at helping understand how the microscope is actually working. We have our laser panel here at the top. We've got an argon laser, so we've got a selection of lines here. We also have 2 diode lasers, and in addition we also have a white light laser. What the white light laser does is that you're actually able to image at any wavelength between 470 and 700 nanometers compared to the argon, which is just your specific lines. So our red lasers currently are not on, so we just need to turn those on in configuration. So we have a 633 and a 561. Just go back into acquire. The sample I've got on the microscope right now are some HEC293 cells, so human embryonic kidney cells, and these have been stained with phalloidin 488. So that's an Alexa 488 dye, which is attached to phalloidin, which will stain our actin microfilament. Knowing that we have this, I know that I want to use the 488 laser line. So if I turn this on, put it on about 10%, then what's nice is that we can actually see the light path. So we can see the light coming, hitting a mirror, and it's being shone onto the specimen. This has gone through the scan head onto the specimen. Emitted light is then going to come back through towards our detectors. So we have our laser line up here. We now have a number of different detectors that we can use. So as I've said before, the PMT, which is a photon multiplier tube, we have 5 different PMTs on our system. We have 1, 3, and 5, which are just all have the same specifications for normal confocal imaging. PMT2 we have for FLIM in particular, and we have a high D detector in position 4, and this is specifically for STED. It's a highly sensitive detector, and it allows us to do gating. For today, we're just going to use the normal PMTs, and as you can see, PMT3 has just been highlighted just automatically, so I'm just going to untick it, and we're going to use PMT1. This is that I'm moving here. This is our emission collection band. So by highlighting PMT1, this is the band that we are going to collect our emission in. To try and get a good idea of whereabouts we want to collect our emission, what we can do is put some spectrum on our detection path. So this is our emission spectrum for Alexa 488. And so I'd say we're probably collecting quite a good window. I can double click on it, and I might just put it to a specific number. It's just good to know so that then when you come later to do another sample, you may want to use the same. So sometimes it's good just to use simple numbers that you're going to remember. So 500 to 550 is quite good. If I now go live, what you'll find is that I can't see anything. And the reason for that is that there's one more parameter that needs to be changed, and that is the gain. So on your PMT, there will be a gain setting, and what this is doing is setting the scale at which your photons are going to be amplified. Just reopen that again. So now if I turn up the gain, now we start to see something. And I'm just going to focus a bit. Always make sure your focus is on fine before you move. Otherwise you'll lose what you found on the eyepieces. Okay. What we can do now is we can actually we can control the brightness of our signal. And what this is important to do this because you don't want to photo bleach your sample. A good way to do this is that you can change the lookup table settings. So there's a button up here. And as you see, we go between, there's either grayscale, false color. So I have currently this in green. It helps a bit to know, you're looking at green. So looking at green is helps make it simple. The other option is a saturation lookup table, which is good to look at whether you're saturating your image. You don't want to saturate your image. If you do, then you have a lot of information in there that you can't do anything with because you're out of your dynamic range. So I know that there's saturation here because there are a lot of blue pixels. If I go live now, there's 2 ways to reduce the signal coming off. We can turn our gain down, and also we can turn down our laser power, which I'm going to do, because turning down our laser power helps with our photo bleaching. You don't want your gain too high, because what gain will do is it will increase the signal, but it will also increase the noise. Okay, so we have an image. Okay, so now we want to look at how this image is actually being created. This is coming back to that scanning part that we talked about in the presentation. So at the moment I've got a pixel grid of 512 by 512. And we can see here this means that our pixel size is 481 nanometers. So one advantage with confocal is that we can actually change this. We can change the number of pixels within our image. So say I change this to a much lower number. Let's go with 64 by 64. And let's go live. So you can see now there's a lot of information lost by doing so. But it is scanning a lot faster. We could go the other end of the scale and increase our pixel number, sorry. And as you can see, this scan's a lot slower. And as I've done this, I've jumped up to 4096 by 4096, and our pixel size has got a lot smaller, so we're at 60 nanometers. So we have a lot better spatial resolution, we have a lot more information in the XY, but it is going a lot slower, and you will get more photo bleaching by doing this. So normally I stick with 512 by 512 when I'm having a look around and I want to find my good self. If I decide on, I'm just going to turn this laser power up a little bit, I'm going to go back into the green lookup table. So if I find the cell I want to look at, what I can do is I can zoom in. And this is going to actually give me better spatial resolution. So if I show you now, I'm going to zoom in. And as I'm doing so if you want to, another way of getting more information is to zoom in. So if I start to do this, if you watch the pixel size here, you see how that pixel size is getting smaller. Also what I'm doing is by zooming in on the cell that I want to look at, what I'm doing is I'm getting more pixels over my area of interest. So this is because I've still got a grid of 512 by 512, but I've got more pixels over these areas that I'm actually interested in. Okay, so when I talked about how to adapt the strength of your signal, there is another way to do it. So we have the laser power, and we also have the gain on the detector. Another way is to look at the speed at which you're acquiring. So at the moment we're going at 400 Hertz, and what this equates to is the amount of time that is spent on each pixel by the laser. If we really slow this down, you'll see it is going a lot slower. It's going to take a very long time to get down to that cell. And what's happening is that that laser is spending a lot more time on each pixel. So it's good to do if you have a very dim sample. So you're going to get more photons per pixel. As you can see, we've got very bright signal now. But it's going to go very slowly, and therefore it's going to photo bleach whatever you're looking at. Alternatively, if we go very fast, you see it's scanning very quickly, but it is that little bit dimmer. So again, normally I'd stick to about 400 Hertz. And I can do now is just capture an image. So it looks okay, but there is a way that we can improve it, make it look a bit nicer. That's what I'm going to do, is turn the gain down a little bit. Okay, the image we just took was just one single capture. What we can do is add in some averaging. So we could say add in a frame average of 3. And now if I say capture, what it's going to do is it's going to take this image 3 times. And then the final image is going to be an average of all 3 of those images. What this does is it gives you a better contrast of an image, because you're going to be reducing the background noise while making your signal brighter, and also it smooths your image out. If you did have a very dim sample, so turn frame averaging off, and I'm just going to turn this down, then what you can do is put on some accumulation. So now if I go capture, what's going to happen is each time it takes an image, it's going to accumulate it. So it's going to add them all of them together. And this can then give you a much brighter signal at the end. Okay, so we're going to move on now to a dual colour sample. So I'm just going to put a different one on the microscope. Put a new sample on the microscope. It's a human pancreatic cell. It's been transfected with some neuropeptide wire tagged to cherry, so we're going to have some red vesicles in there. And then also I've got my phalloidin 488 again, so we're going to have some green actin in there as well. I'm just going to zoom out, and I'm just going to look with the green first of all. Okay, so then we can see in this sample we have our green actin inside our cells. Okay, and now switch screens back. Okay, so like I said, we've also got some red in this sample, got some cherry, so I'm going to turn on the 561 laser. You can see our laser line comes up here. And I'm going to choose to use PMT3. As I've said, I don't tend to use PMT2 as it's for phlegm. A few things with this, first of all, is I'm going to get the spectrum up for the emission. And this is for Alexa 568, but it will be similar to cherry. So I can think about where I want my emission band to be. So if I activate my PMT, and I'm just going to move it so it covers a nice peak there. One more thing I'm going to do is change the lookup table to red. It's a lot easier to see than blue. Okay, and finally, do you remember from last time, we do need some gain. Okay, so now if I go live, okay. All right, great. So I found some cells which have got some vesicles in. So if I just stop, I'm just going to zoom in. On this region and go live. We've got our vesicles in red. We've got our actin in green. So same as we did before, just look at the signal from both. You will need to have different laser power and different gain for each fluorophore. So let's just have another look at that. Red, get a little bit brighter. Okay, and I'm going to choose to put some averaging on. Let's make it look a bit nicer. And now if I capture my image, we're going to see both are taken at the same time. What I can do is press on this button here, and that gives me an overlay. So I double click on this now. So you can see that some of the green signal's gone a bit yellow, and that's because they're both in the same place. And then we have the red. Now, with what I've taken here, if you have a look at that red, it is actually a bit saturated. So I might go back, live. And let's have a look at this one in particular. So I think that's actually, might have the gain on a bit high. That looks a bit nicer. So now if I go capture again, it looks a bit better. Okay, so that's our dual colour imaging. So now we're just going to have a last look at the pinhole. And to do this, I'm just going to change sample again. Okay, so I've just changed back to my single colour sample. So I'm going to turn off my red laser and I'm going to turn off my PMT3. So if we zoom out and go live, good. So okay, so I'm just going to crop in on an area. Okay. So the last thing that I just want to show you comes back to the pinhole, which we've spoken about through the presentation. As I've said, there is a pinhole in place in front of the detector, and what this does is it's minimizing the amount of out of focus light that's coming through to the detectors and giving us this nice sharp image. And on a confocal, you can change the width of that pinhole, the width of that slit. So I can show you this here if we go live, and if I reduce this pinhole down, you see how we start to lose light, gradually so much that you can't see really anything. What we could also do is increase. If we keep increasing, you see how, for one, it's getting very bright. So we just turn this down. Also what's happening is you're getting this blurring, and that's what we've talked about with wide field illumination. So when there is no pinhole, then you collect all of this out of focus light and you get the degradation of your image. So if we go back to an area unit distance, which is what we're used to, you see that difference there. It's a lot sharper, and you get that typical confocal image that we're used to. So thank you very much for your time. If you have any questions, please let me know.
Thank you, Alison, for that talk. That was really interesting. Like I said, if anyone has any questions, please pop them in the questions box. We've already had a few questions already, so I'll get started. Can confocal scanning microscopy be used for the quantification purposes to say, look at the differences in the expression of proteins?
Yeah, I mean, it can do. I think it's something that is possible to do, but you just need to be wary of knowing that your different cells are going to have different expression. And intensity is something, quantifying intensity, as long as you try and keep your settings all the same. There are options with certain detectors that you can do, so like single photon counting or photon counting, which is more quantitative than just calculating intensity. So it is something that's possible, it's just something to do carefully.
Okay, that's great. Quite a few people are asking whether or not this will be available online. Yes, we will make a version of this available that will be free to watch, and it will be uploaded to our webinars section of our website in the next 24 hours. And just to carry on with a few of the other questions, what is the speed difference between spinning discs and scan confocal?
To be honest, I'm not sure. I can tell you on a confocal, so I've not used spinning disc very much personally. On a confocal, to take, say, a stack of a single mammalian cell, you probably take you about 15 minutes to take that at the proper sampling rate. On a spinning disc, I'm not sure, but I can try and answer that at a later date or online later.
So we have got quite a few questions that are coming in. So any questions that we don't manage to answer in the webinar or are highly technical, we'll make sure that these get answered and the answers get put on our website along with the webinar. Another question, Alison. What is the best way to deal with autofluorescence in terms of separating it from the real signal that you want to detect?
I think with autofluorescence, the most important thing at the beginning is with your sample prep. So trying to, sometimes using things which will quench your autofluorescence, making sure obviously there's no phenol red in your media, and things kind of along those lines. With autofluorescence, I would just try and focus on not having your gain too high, because that's where you might detect it if you have your gain too high on your detector. They're kind of the key things I can think of.
Can confocal imaging data be used to quantify structural features such as dendritic spine density, or is the Z-plane resolution too limiting for that?
Possibly it could be too limiting, dependent on the thickness of a dendritic spine, which I'm not sure. But since your resolution in Z is about 500, a bit more, then possibly. But with doing confocal, if you did pair that with deconvolution, deconvolution can help to increase the resolution and make your images a bit sharper, so it may be able to help in that situation.
Okay, how can you improve the resolution in a Z-stack?
So in a Z-stack, so if you can increase it by having more pixels. Bearing in mind, obviously, you will have more, it will take a lot longer. I would say that if you take a Z-stack, and as long as when you do take a Z-stack, it is very, very important that you sample correctly, and you can find out how to do that using the SVI website, which has something called a Nyquist calculator. and that can give you an idea of the sample step size that you need to use, and also the lateral pixel size that you need to use that will give you the best sampling rate. If you do this, again, you can use deconvolution, and that can help with the Z resolution.
Okay, thank you. Are there optimal laser powers for each laser, or is this very much sample dependent?
I would say it is very sample dependent. The laser powers are, it's a percentage of, so we have our argon laser on 30% in the back and our white light on 70% in the back, and then on the front we change our percentage, but it is dependent on the lasers for one. So when we had our upgrade, our actual percentages changed, and we had to kind of get into the mindset of using a different percentage and thinking it was brighter, but actually it wasn't, because it is just an arbitrary percentage. And yes, it is quite sample dependent, so I wouldn't think, well, I used it at 20% on this microscope, then I should use it on 20% on the next microscope. Or say day to day, sometimes if you have a bad sample, then you may need to change your laser power.
Is white laser tunable to a particular wavelength, or does it excite at all wavelengths?
It is tunable. It's a tunable laser, and you can use it at an excitation between 470 and 700. So I think that I might need to give a bit more of an in-depth answer, I think, online.
Okay, that's not a problem.
If that's okay.
The next thing is, what's the difference between line scanning and frame scanning?
In which context?
I don't know. We'll have to ask if the person who asked that question could maybe give a bit more information, or perhaps that's one that we can deal with in the future on our website. I'll provide an email address, so if anyone has any further questions or follow-ups, they can email me and I can pass these on to you. How is it best to improve the resolution if you've got a live, thick sample? Are there any particular tips for improving the resolution for that?
I'm not sure. It's not something that I have done a lot of with thicker samples. So again, that might be something that I can have a look into and get back to you on.
I suppose this is maybe slightly related. Do you know the range of depth that a confocal microscope can scan?
It depends on the lens that you use. In terms of like working distance of lenses, if that is the question, then it depends on the working distance of the lens that you use. But obviously, the higher magnification you go, then the shorter the working distance.
Another question, what gain should you use, and can you go too high?
Yes, so it is kind of dependent on which system you're using and where you're working, to be honest, because everyone has their own rules, but we do tend to not go above 800 on our PMTs. You know when you're going too high because the background will increase, and when that starts to happen, then you're better off turning down your gain and increasing your laser power. So yes, it's definitely possible to go too high, and it is something to be careful of. If you can't see anything much, then you can't see anything, and cranking the gain up too high is just going to give you a lot of background.
Okay. Is there any issues with multicolour imaging, and is bleed-through a problem with this?
Yes, it can be. So there is, on most, I'm sure on all confocals, there's the option to sequentially scan your image, because often green and red are the two that you use. Then if you can see that the spectrums overlap, so you are going to get bleed through from the green into the red if you image both at the same time, but it is possible to sequentially scan one and then the other, which will minimize this. There are also tools to be able to spectrally unmix, and this is something that is possible. It's just, again, it's something that needs to be done very carefully to make sure that you're doing it correctly, but that is possible for things which are quite close together, such as GFP and YFP.
We'll just bring it on to one last question. Noise to signal ratio is of crucial importance for any imaging, and it largely depends on the specimen. In scenarios when the fluorescently labelled protein localizes to two structures within the same cell, one which is very brightly labelled and one which is very weak, is there any way to optimize the imaging without having saturation?
I mean, obviously saturation is very important, that you don't saturate. If you do a sequential scan, then you can try and optimize for each single image. There is, depending on what your setup is, the high D detector does have a setting on it which is called Bright R. I'm not sure if this is the same for, say, grasp detectors as well, but it's a setting where it will make things in your image brighter that are dimmer, and dimmer which are brighter. It's not always good maybe to quantify things from that, but it can help to try and get an idea about where things are in your cell. So yeah, so that is an option.
Okay, so I think that brings us to the end of today's seminar. Thanks again, Alison, for a very illuminating presentation and a wonderful discussion. And thanks also to our sponsors, Leica. And finally, thanks to the audience for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours. And all the previous webinars are also available to view on there. You can also see on there our upcoming webinars, including one from Alison tomorrow, discussing how to choose the right technique in microscopy for your scientific question. And that just leaves us to say, so until next time, good luck in your research and goodbye from all of us at Leica and Bite Size Bio.